Okay, well, as people are starting to pop on slowly, I will go ahead and introduce the speaker today. So NIF is very pleased and proud to welcome Dr. Tom Hawkins, who's a research fellow in the Cell and Developmental Biology Division of Biosciences, Faculty of Life Sciences inside of the University of College London. So he was, he got his PhD in neuroscience in the University of College London a few years back. Primarily his work entailed working with peripheral nerves and the various ways that those are destroyed and can be brought back. However, one of his more recent and dare I say passions is to really work out a neuroinformatics resource for the zebrafish anatomy, which I dare say is lacking to some extent at the moment. So I think without much further ado, I would love to welcome Dr. Hawkins, and please tell us about zebrafishbrain.org.
Thanks very much, Anita. I guess I should just go on. I imagine Jonathan might be joining us soon, but should we wait any longer, or should I just go on with it and he can join in?
I think we should probably just start. I'm sure that he'll be on in a couple minutes.
Okay, so thanks all for coming, and like Anita said, I'm going to tell you about zebrafishbrain.org. Jonathan sent me an email to ask me to do this because he figured it would be useful for NIF to what we're doing. Sorry, just I can get a pointer somehow, can I? Yeah, like that, right? Is that a pointer? Yeah, okay, right. Just wanted to know I'll need it later on. Sure, it's all a bit new to me. So, right. So I'm just going to say first of all what I'm going to talk about generally, how the talk is going to be structured. First of all, I'm going to talk about zebrafish brain, sort of describe what it is, what our objectives are with making it, and its current structure. And also, really, one of the key things I think Jonathan alluded to in his email was that ontology would be a really important thing for us to discuss, and work on combination between and ZFIN as well, the Zebrafish Information Network. Then I'll talk a little bit about where the data that's populating the atlas is coming from, including where our transgenics come from, and how we're kind of gathering the data and processing it, etc. I think I'll try and focus more on this than this, but this is quite they're nice pictures, so I think you'll all enjoy those, hopefully. Okay, so I might have time, I don't know whether I will, to talk a little bit about how we're kind of working describing neuronal circuits. Okay, so one of the key questions that people often ask, well, I guess is sort of a key question generally, is why do this? And I think Anita quite succinctly kind of alluded to that, which is that really this kind of resource does not exist yet for zebrafish. There are plenty of resources in other species, and it's time we had one for zebrafish, really. And really the reason is that you want to go from, in order to be able to go from kind of simple molecules to understanding behavior, and you really have to go through neural circuits. You have to understand what the neural circuits that derive the behaviors. And a zebrafish community doesn't really have this kind of source at the moment. All we have is the great canon of literature on various different species, not just zebrafish, but other species. The sort of Z anatomy browser, which is a purely text-based thing. So we need something better than that. And really that jars a little bit with the fact that I'm sure all of you know that zebrafish are kind of considered to be particularly beautiful because of their optical properties, and we have many transgenics that are available, and you know, the actual accessibility of the embryo during development from single cell to swimming larva, you know, all in a dish, is kind of, it's kind of ridiculous that we don't have something which can communicate the anatomy more clearly. They're also very simple species, especially compared to mammals, and particularly when you think about their kind of behavioral repertoire. Fish are fairly straightforward, really. I mean, they don't do complex social interactions so much. I mean, they do schooling behavior to a certain extent, but nothing really close to even mice. So I guess that's really is why we're doing it, and this kind of hopefully shows where we're going. I'm sure lots of you have heard of this phrase connectome. It's kind of becoming a thing now, and this is a little diagram here showing the it's from the Sidney Brenner paper, kind of seminal paper, showing that showing the brain of the worm, as it were, all of the different brain cells of the, which was done using, oh dear, someone can't hear me. Wendy, should we wait a second, see if we get that sorted out?
That's all right. I'll do a private chat with Wendy to make sure her configuration's okay.
Thanks, Rita. Okay. So this exists in the worm, this connectome, a complete mapping out of all of its the worm's cell sort of neuronal pathways, and this was done using serial section EM very laboriously over 20 years ago. And really, where we want to get to with zebrafish is to get to this point. Obviously, we're a long way off, but if we can understand that, we can start to understand how to get from molecules to behavior. And really, the reason I mentioned that really is that C. elegans really shows the power of this. So now with C. elegans, you can do genetic screens, etc., and knock out individual neurons or manipulate them, make transgenics that can allow you to manipulate particular neuronal activity, etc., the physiology of particular individual neurons. And you can understand the consequences of that for the behavior, but also for kind of other components circuits, because we have this connectome. So we need to get to that in order to really progress. And it seems like to me that being a vertebrate, zebrafish seems like a good place to sort of start working on connectomics in vertebrates. So yeah, it's obviously a long way off, and everything that I say today is very preliminary. What we're making is all very early. I'm communicating this at a very early stage of development, so just to see if I can get interesting feedback, and any kind of any feedback is obviously welcome. So our aims really very simple. Describe the neuroanatomy of the zebrafish. Oops, maybe that flipped back. I'll go on again. Okay, and we want to collect and make these some key transgenic lines which describe neuroanatomy nicely. We want to communicate this information in a kind of intuitive way, where possible using 3 dimensions, because lots of people using zebrafish use whole mount preps, and we don't really want to use sections, etc. We want to use sort of 3D presentation wherever possible, and we're kind of trying to do that. We want to kind of act as a repository for this data, for any neuroanatomical data, and really we also want to sort of form a catalog of neuronal morphologies and their kind of and describing their connectivity really. So that's what our aims are. So a good thing to think about really at this stage is who are the users, who are we kind of aiming to build this for? Obviously, it's the sort of zebrafish community, but also more widely, I guess, people who want to know about structures and homologies of structures in zebrafish from other species. So really, like I said, we have no data really so far on on any users we have, because we don't really have much of a resource yet. So we're kind of imagining that the users will be split into expert users and sort of more novice users. And I've maybe slightly irreverently included a few pictures of who you might imagine these could be. So an expert would be someone like a Hal, who would be a neuroanatomist, or a neuroscientist, or someone who has a good amount of knowledge about neuroscience, and they might use this resource as an archive for their data, and hopefully they'll be able to be recruited by us to edit the resource and really provide information to us and to the community. And then novice users might be these, for example, molecular biologists, or drug screeners or pharmacologists, people who not necessarily don't really know much about zebrafish, but are working with zebrafish. They might be very new to zebrafish, come from other species, and they really will require information on sort of homology of structures, where they are, if they're wanting to work on a particular structure. Also, having information on markers for particular structures is very key. So that's what we think about users. We want to try and obviously cater to both, and we also particularly want to tap into the knowledge of the experts in the field. In terms of numbers, we're thinking probably the number of kind of expert users is actually not going to be that many. There aren't many really expert kind of zebrafish neuroanatomists. I'm certainly not an expert in all of zebrafish neuroanatomy. There are quite a lot of people who would be expert in kind of a comparative anatomy of teleosts, which is kind of the same thing really, and we want to try and tap into their sort of knowledge as much as possible. But the numbers of these is going to be in the tens really, and the number of these is probably going to be more likely in the hundreds. So that gives you an idea about how many people we're going to be serving. Okay, so what do we do? It's been a bit slightly formatted here, but it doesn't really matter. So this is essentially distills down what we do. We take transgenics or aminos or data sent from other labs, that's supposed to say, and just take images from it. It might be a stack that we're using that comes from somewhere, or it could be I've lost the pointer there, but it doesn't matter. It could be a stack, or it could be individual images. We annotate them, and we use them for descriptions. We annotate them using a controlled ontology, saying this bit's this according to a particular ontology which is provided by the zebrafish information network, ZFIN. And then we incorporate them into tutorials as well. These get that.
Okay, the pointer on my screen is over on the left, so maybe you should move your mouse right. But I actually have a question for you here, if that's okay.
Yeah.
Okay. So do you have a large image repository in the background which you'll be accepting data for of this type, or do you, are you looking for that? Do you grab those large images from elsewhere?
We get people, will send us data, and we have a large repository of images, and we're slowly feeding them onto the our database as we're making it. Really, it's not housed on the web or in the database, as it were. We're trying to sort of distill down the best data on particular structures, really. So yeah, if that's what you mean, does that answer your question, or are you what?
Yep, that pretty much answers my question. Thank you. I just wanted to, if for example I run across somebody who has very large stacks of images of zebrafish tomorrow, would I be able to direct them to you as a repository?
Yes, but when I say repository, we're not planning on we'll certainly archive the data and keep it, but it might not be on the web. We're not going to display it like probably it'll become clear as I go on. I think maybe I should just keep going, and it'll become clearer what I'm talking about. Maybe. I can't seem to get that pointer to come off the side of the screen. I can make my screen smaller or bigger. Can you grab it and push it across? I'd like to.
No, I really can't grab it. It seems to. Maybe I could make it go away and come back. No?
I tried
Nope.
that.
All right. Oh, I've grabbed it and I've moved it onto the screen.
Got it. Okay. So I mentioned tutorials. So we use some of these images to kind of populate a tutorial. This is represented by this sort of funny little schematic here. So the annotated images, which we annotated according to a controlled ontology, populate a tutorial, which is effectively like a page on a particular structure, which is also derived from a controlled ontology. And then all that goes to the web. And like I said, this is all really in development, and that's a kind of key consideration because it's early days. So I wanted to, at this point, navigate away and go and show you it. So I'm going to try and do that. Now I go to share my screen. We'd share my desktop, and I think over here, and I'm still looking at the Olympics here. Okay, so here is the zebrafish brain atlas. This is how it stands at the moment. This is our homepage. You'll see on the top here these tutorials that I was talking about, and various other things. There's a nice image to show people, so you've recognized it from my first slide probably. At the moment, these tutorials are just arranged in a kind of slightly not so much haphazard. They're actually very alphabetical, but they're alphabetical with systems followed by structures. But our idea is for this to be in time to be an actual kind of three-dimensional navigational sort of brain, as it were, so people can move around in 3D, touch a structure, and then it takes them to a particular place. So they might know they've got expression somewhere or whatever. But for now, let's just go to one of these tutorials. My favorite tutorial, Habenula. It's really our favorite structure in the lab, so we're kind of expert on it. And this shows you what a tutorial looks like and what these tutorials are like. And this is the most developed one as well. And really, it just has a title with a Habenula. It has some synonyms here. Synonyms are very important, as I'm sure you know, in ontologies. It has a little slideshow showing you some nice sort of key data. There are 3 sections: introduction, neuroanatomy, and development. And sort of introduction is what it is, and has maybe some schematics or such like, along with a little slideshow. The neuroanatomy has more detail on a particular structure. It might have information about sort of where it is, ways of staining it, ways of marking it, cell morphologies that are in there, and schematics describing its structure, more cell morphologies or axonal morphologies, etc., and sort of information about its connectivity. Then the development page, which is fairly thin on this particular one, has some information about its development and its structure. And what I really want to draw your attention to is on the right here, this kind of dynamic menu system we have, which populates according to whatever structure we're on. And there's a few things on it which I want to point out. So it has a status, which is currently every tutorial we have is in development, but in time that will probably become reviewed or such like, or we have other statuses, so people know that it's maybe a little bit more complete. It has, most importantly, a list of authors, and that's kind of a key thing for when we're trying to elicit input from other people in the in the field that they will be able to have citation here, so that they can well, not a citation, acknowledgement here, so they can take ownership of it, kind of thing, and also be able to put a CV, etc. You know, I've contributed to this, so you know that's a key thing. So we need to have authors up here, and that's regulated by somewhat by our curation of it. So we make sure that if someone wants input, then we give them a login and they'll appear there, whenever they make any kind of input. And there's this related regions thing, which is really a readout of the ontology behind the whole thing. It says it's part of the epithalamus and it comprises these other parts. You notice the epithalamus is a link because we have a tutorial on the epithalamus, which we can go to, and we can go back to the habenula, which is part of this. But you notice in the epithalamus it also has other related regions which actually should be under here, but have somehow appeared up here. But the epithalamus is part of the diencephalon and it comprises the habenula, pineal complex, and etc. other places. But we can go back through to the habenula there. So it gives you an idea about how this is going to build as we get more tutorials here. It's more browsing here. These other related regions are just related for other reasons. The fasciculus retroflexus connects to it, etc., etc. So there are reasons why they might be related, not just part of the ontology, although that should be included in the ontology, but is not currently, and that's something which we're going to be inputting as time goes on. Okay, so we have references, and these are the key references. For a while we had a full list of references here, but it made this menu so big, so we've narrowed it down to only key references. And if you click on this view all thing, you get effectively a sort of annotated copy over here, which are these key references, and then there are some other references which are used in this, and all of this is links to PubMed, etc., and has some a kind of commentary really from us on particular papers that are key for particular structures. Okay, so we go. It doesn't really matter where. Then a similar thing with transgenic lines. We have key transgenic lines that mark structure here, but the other transgenic lines that are involved. So there's actually only one here. This has details of the transgenic lines. Also has details of where else it's expressed, and this is all just in order to kind of make the whole thing a bit more kind of interactive and clickable. Then this section here, media, which is very much under development, is intended to contain all data on a particular structure. So when we've got annotated images on a structure where there is an annotation for that structure, it will be included here in this media section. And at the moment, this is, as I say, under development, but you end up with some annotated images. There will be also legends here for these particular images, and these may not have been used in the actual tutorial, but they're extra images that might be interesting for people, and that will just build automatically as we annotate structures. And these shows you an example of some annotated structures and the way we're kind of annotating them as well. So with just sort of little dotted lines and such like, and we have arrows and other things, so dotted lines and letters. And then there's just typical kind of glossaries here down here, which we're kind of building up slowly as well. We also have some videos available here. I don't know whether, here is a video on this particular structure, which we can play here. And this plays using a Vimeo sort of plugin. I don't know whether it'll work very well. Not terribly well. Yes, it does. Not bad. Okay. So it's not the best video we've got, but has some legends here. And our idea is really also to present 3D things, as I said at the beginning, as much as possible. And here is an example of exactly that, right? There's a structure which is being presented in 3D. It's just as a simple plug-in movie. Our intention would be in the future, I guess, to have ways in which things can be much more interactive. But for now, I think it's going to be plug-in movies until we get that sorted out. And we are planning on collaborating with a lab, a computer science lab, to get that sorted. So I think that's pretty much all I want to show on this. I'm going to just go back to the home page and show you a few other things. So that media section, here we can actually view all of the media here. So not just the ones which I showed you, but some more, and some more movies here. There's indeed a nice little introduction to the zebrafish brain which plays here, which has my dulcet tones narrating it. And as you can see that quite a lot of these are quite common to the ones we just looked at. Okay, so that's me. And there's also a kind of guide which shows you around the tutorials and says all what I was talking about. And that's all on the go and sort of up and running. Once this bit is up and running, we plan to upload a lot more data and get that whole bit much more populated, and then really work on populating a lot more of these as well. But as you can see, we're not doing too badly. A lot of these are quite thin. Battery bulb has quite a lot of detail, quite a lot of text. This one, there are quite a few pictures down here as well. That's probably a bit of a hangover from the time when we couldn't integrate text with figures. Okay, so there are a few other things. There's a sort of how to use guide. There's some information on status. That goes to media again. This community page is kind of important. This one is obviously interesting to you probably. Has information about our sort of policy with regards to ontology, which I'll talk about in a minute, so you don't really need to read it now. I think that's it for now. Here again is another way of browsing different regions, but don't worry too much about that. I'll go back to the presentation, which is on here, hopefully. Hopefully that works. Yeah, always gone back to the beginning. I'll just flick through. There we go. Okay, thank you. I think maybe Anita did that for me. So I had these slides to just in case I couldn't get to the web, so don't worry about that. This just talks through the tutorials. I already did that. And these are the dynamic menus that I'm talking about. Here are another couple of examples, which are slightly older formatting when I was on about using references, the whole list here. And this has changed slightly, and we've rearranged the order, but basically it's the same thing. This goes from forebrain to telencephalon. You can see there's a couple of authors on these, and that's probably all you need to see there. So really, because I did it on the actual thing. So going back to ontology, I said I was going to talk a bit about that, and I'm keen on getting any information that you want to provide me on. I don't know how NIF, NIF using kind of curating ontologies, but we run on the ZFin anatomical ontology, and it's an oboe format ontology. This, some of you might recognize, is the oboe edit kind of view of some part of it. In fact, it just is sort of the stems of the nodes of stemming from the brain and the various relationships, just to sort of demonstrate it. At the moment, this ontology is definitely a little bit sparse, but we're hoping to kind of increase the amount of data there. And there's not much kind of comparative information. I don't quite know how that's dealt with very well, or how one deals with that indeed in ontology. But we're keen on sort of having input into this. I don't quite know how that would happen, but we're certainly keen on developing this in collaboration with anyone who wants to collaborate with developing it, really. Okay, at the moment we kind of have an update system whereby ZFin publishes their oboe format ontology once every month or thereabouts, and we kind of update what our kind of database implementation of that ontology in a one-way fashion, but we kind of are keen to feed back on that. And at the moment, there's a kind of slightly clunky way of feeding back information to ZFin, but it's kind of coming along, hopefully. And we had a big meeting recently about it, so hopefully that is going to make things progress. So yeah, like I said, here are another couple of areas of the brain, just to show you. I think I was showing this to some other people just to show how these ontologies work. But it's sort of pretty pictures, and really we plan to make our assistance in the curation of this a kind of a major output of what we're going to major output of our efforts, because I think it's a key thing to get the ontology right. and kind of keep it active as well, actively being kind of curated. Okay, so some details about database for anyone who's interested in techie stuff. This was a slide done by my developer, who is Stephen O'Connor, or our developer, and it says lots of details about how it works. We have a web-based admin system, so you can actually edit the tutorials online. I'll talk about that in a minute. On the web, so that's the idea, is that people that allows people to, us to solicit input from other people in the community. Any uploaded images are resized and compressed. Originals are stored untouched. We have a backup system. At the moment it's 8 terabytes, but it's flexible and can be increased in size easily. We have all of the text data is in MySQL file. We use Vimeo for movies. Like I said, ontology is on Zfin, and we kind of simplify slightly the ontology just for our database sake, just based on direct physical relationships. I had a little bit picture of different relationships in that ontology slide, but that's the way it is. And we have developed a hierarchical browser that literally goes through sort of forebrain, midbrain, blah blah blah, and allows you to browse through them in a text-based manner. But what we want to do is a sort of more of a 3D type thing. But for now, we do have a hierarchical browser we can use. But like I said, for simplicity at the moment, we just present a list of tutorials, but that will change as time goes on. Okay, that's a bit of that. Not my particular field of expertise, it has to be said. Okay, so yeah, as I said, I was going to talk a little bit about our objectives, the current structure of the resource, a little bit about ontology, and now I'll talk a little bit about where the data comes from and how we gather it, process it, and present it. So here's a slide showing all of our transgenics. So some of our transgenics, not all of them. Apologies for anyone who's red-green colorblind. We are now trying to implement a policy of using only magenta and green, and no red and green where possible. And all of these have actually been changed. And this is an old slide, so I'm very aware of accessibility issues, but it's a bit of an old slide and I just never changed it. But this hopefully gives you a bit of a flavor of all the different kind of transgenics that we have available. And this is the brain from dorsal view, lateral view, ventral view, lateral view, dorsal views, and various other views. And it's really just to show you a kind of potpourri of the different expression patterns that are available. And all of these we have in our facility. And so far we've looked at about 30 different lines. So this is only a selection. From different places and actually different types, most of which are GFP lines. We're trying to move towards using transactivation systems, GAL4, etc., and also to move to what I call standardized lines, which implies kind of characterized promoters and such like. So we don't end up with just a load of markers for particular structures in the GFP lines that it might be from enhancer trap screens where we don't know where the enhancer comes from, etc. We're trying to sort that out and ensure that everything can be repeated nicely and can be recapitulated again. So, right. So yeah, transactivation. This slide also nicely illustrates our primary counter stains, which are in red here, tubulin, which just marks sort of axon tracts and a bit of neuropil. And also we use this stain in cyan in this little image here, which is SV2, which just stains neuropil areas only. And so the two of them are quite complementary actually. And in fact, this is also very complementary. SV2 is complementary to nuclear staining, which we use quite a lot. So I think maybe that comes up in a minute. Okay, so transgenics. So we've committed ourselves to our funders between now and 2014 that we're going to image and incorporate data from 100 transgenics, not 1,000, 100 different transgenics. And we've done about 30 so far, as I said, in terms of gathering data. We haven't incorporated that data into the atlas yet, but we have a big pile of data from these lines, close-ups of different areas of the brain in different lines and with different counter stains. For the additional 70 or so that we need, we're going to raid various different existing collections of which we've started to. Here is a line from Herwig Bayer from UCSF, was UCSF, now he's in Germany. We have a number of those, which are actually GAL4 lines, which is nice. And like I said, moving towards transactivation. This is actually, I think it's probably KIDA you're seeing here in the immuno. We are undertaking a small-scale enhancer screen here. I'm trying to move away from enhancer trap screens, but we probably need that to get to our sort of target. We're converting various different classical constructs to become GAL4 type constructs, and that's going to be Kate Turner in our lab is undertaking that part of that. So these are, when I say classical constructs, these are constructs which use defined promoters and might have been used to make a GFP line, but now we're going to be using them to make nice GAL4 lines. Andy Simmons in the Clark lab, actually in King's College. So this, I didn't mention at the beginning, but this project is actually a collaboration between King's College London and University College London. And Andy Simmons is working in John Clark's lab down there under the same grant. And he is in the process of doing back transgenesis to make constructs to try and mark different neurotransmitter systems, particularly neurotransmitter systems in the brain, targeting their receptors, also enzymes and the neurotransmitters themselves where possible, and making these constructs. He's in the process of making the constructs. He's made about 30 constructs so far, and we're waiting for the transgenesis to happen, mostly using a TOL2 transposon system. So what else? So we have a few character enhancers that are coming out of a couple of other interesting screens that are going on, one of which is called NeuroX-Cis, which is examining cis regulation on the X chromosome. And this little diagram, this little couple of things down here indicates that. And this is a locus on the X chromosome, which is being characterized and a GFP line produced. And then it's being converted into a GAL4 line. This CAL4 is actually a version of GAL4. You can see hopefully that this expression pattern just about, you can see two blobs here, right? And there are two blobs here. So you can see that this expression pattern is certainly recapitulated in the GAL4 line, which is really nice, showing that it's a very stable kind of characterized enhancer or sort of cis-regulatory element that could be used to drive expression of whatever you like, and particularly in this case using GAL4 we can transactivate various things using US lines, which already are starting to become more available in zebrafish, which is great. That process is happening. It was a big kind of collaborative grant between various people. Ben is in our lab, but then between the Ballyqueef lab in France, Australia lab in Germany, and the Becker lab in Australia, and that's ongoing and finishes sometime this year. And we should have, I think, probably at least 15 or so characterized cis-regulator elements that are coming out of that. Obviously, the mission for this was really to understand cis-regulation on the X chromosome, but our kind of feed out of it is that we end up with lots of nice lines which are stable, characterized cis-regulator elements which we can use to mark particular parts of the brain. So that's one other source of transgenics that we're getting. Like I said, we're doing an enhanced trap screen. Someone want to say something? I heard a noise. I think that just was switching on.
Oh yeah, I was going to ask a. Oh, sorry. I was going to ask a question about your transgenics and whether, how are you going to feed those back to the community, or, are you making them available, since you're making them, are you going to make them available through ZFIN, or what's the policy there?
Right, sure, that's a good question. So I said that we're definitely going to raid various existing collections, and a lot of these we're going to try and stick to ones that are available through Zerk, and the European Stock Center when it gets going. It's in the process of getting going. So we'll submit them to the stock centers for sure, so they can easily be available, and we'll also hold a stock here. So People in Europe certainly can get easily from here, and in the UK. And like I said, we'll submit to Zerk as well. So anything that we put up on the web will eventually be easily accessible by the community through Zerk, probably most likely, or through the European Stock Centre. Obviously, that leaves out a lot of Asia and Australia and such like. And I think they're probably going to be working on stock centres themselves. And so hopefully in time, these things will be distributed all over the world. So that really hopefully addresses that. Is that okay?
Yes, thank you.
Okay.
I just keep muting myself because...
Yeah, sure. It makes it easier for the sound. That's understandable. So yeah, we are undertaking a small scale enhancer traps screen here, partly for fun, but also because we do need to make sure we have enough lines. And so this is with a collaboration with a chap called Masa Tada, who's a lecturer in our department. And this is a sort of flip trap type construct with flip recombinase sites here. Are they flip? I can't remember. Actually, I think these are recombinase sites, not flip recombinase. I don't know why I've called it a flip trap. It doesn't really matter anyway. The idea is that you screen for GFP and you can remove the GFP and you have GAL4. This is a version of GAL4, by the way. This CalTA4. I don't know whether people know that. Kind of key considerations: which basal promoter to use, and we've tested a couple of different ones. We've got some F1 lines. We want to check that this works first. We're going to be doing that soon. We're right at that moment now, testing that. Then we want to scale up production, hopefully, and end up with certainly more than a handful of lines from this process. But the issue with that again is that you end up with having to then characterize what the regulatory element is by mapping the integration site, etc. And it can be troublesome. And so I'm kind of moving a little bit away from enhancer traps, but this kind of enhancer trap where you end up with a GAL4 at the end of it is probably more useful than just a GFP, of which we have quite a few already. So what do we do with these? I think I've sort of mentioned somewhat on that slide of all of the different transgenics, but we double stain them with different counter stains. So acetylated tubulin, this is a time series of acetylated tubulin staining a lateral view of the brain of the fish from 24 hours to 5 days, and you can see how the complexity really increases, goes like that, and then like that. This marks axon tracts primarily, and it was kind of characterized by my boss Steve when he read a paper about how you get an initial scaffold of axon tracts which is then built upon, and it's a nice marker, though the antibody unfortunately doesn't penetrate very deeply into the brain, so it tends to mark more of the outside. Another antibody we use is SV2, I mentioned earlier, which is this neuropil one, and it's nicely complementary. And so the idea is that these two help locate expression of a transgene in a kind of anatomical framework. And all of the data that we get from this is archived carefully. Hopefully we can use sort of morphing technologies and such like to kind of to overlay different expression patterns on top of each other, which I will talk about in a minute. I don't think I'll talk too much about this slide. I left it in maybe from an earlier presentation. It just was a movie playing through a nuclear stain showing that it's complementary to SV2, but it's not very interesting. We use high-end kind of objectives and nice confocals. We're just about to get a new one, which we're all very excited about. And we use high-NA lenses, so we get nice Z resolution. And we've been using this XUV software, which is quite a nice piece of software for 3D stitching. So the brain doesn't quite fit in the field of a 25x lens. So if you do two stacks, you can then stick them together using this 3D stitching software, which I recommend to anyone to use. The XUV is freely available. And this is an example of a neuron that's been stitched together. Lots of different stacks, and they don't have to be very carefully They have to be overlapping, but they don't have to be carefully arranged, as you can see from this. And this is all developed by the Driva lab. We work quite closely with the Driva lab, and they're actually in the process of making this nice new tool called Vibes, which we're currently testing, which is exactly what I was talking about with regards to morphing data on top of each other. So we're hoping to use that to populate, sort of look at our data and populate the database. So this is a bit of a blown up version of the slide I showed earlier about the sort of structure of what we do. and how we gather and process it, but hopefully a bit more detail. So we have lines in our facility here. We import them and we image them, but we might get data from external sources where other people have done this. And I mentioned after Anita's question about how actually lines end up being exported from us to Zirk in time, hopefully. Once we've imaged, we end up with a stack, which we archive carefully, so it's traceable. We can go back to the original data if we need to. We then examine it using 3D visualization software. We take snapshots, and we segment. Ooh, the sound just went a bit wacky. I think Jonathan turned on his microphone, maybe.
I got a reduce echo button. I was just curious. The new 3D stacking software that you have, I assume is similar to ZStack, but with several improvements.
So I can barely hear you.
Can you hear me better now? Or?
Yeah, that's better.
Okay, I was going to type. So the 3D stacking image software, where you take the 2D images and then stack them together, I would assume it is similar in many respects to a ZStack, but it looks this is a far more improved version.
I haven't used ZStack. But probably, yeah.
What was the name of the product that they're working on?
Vibes. Yeah, I'll talk about that in a minute.
Okay, excellent.
Okay, so I'm still feeding back, but I think John's going to turn off his mic. Great. Right, so yeah, then we look at the stacks using 3D software. We take snapshots, projections, and movies. We're planning on sort of segmenting the data where we can, but it's quite a troublesome manual process, and that Vibes software that I was talking about will hopefully help in this regard, and I'll talk about it in just a minute. We end up with an output of some images, movies. These are uploaded to our database, and they might be narrated, or if they're a movie, or they might be annotated using this annotator. This is actually a movie of me doing it, but I'm not going to try and play it because I think it probably won't work. I could show you, but I think I'll pass on it. It's pretty clear. You just draw around stuff, and you can choose colors, and you can do freehand, you can do arrows, etc. And you have to select when you actually annotate. So this list is a drop down of regions. So if I wanted to annotate the Herbenuli, which have been actually annotated down here, you have to say these are the Herbenuli, and that list is the ontology, from the ontology. So automatically you're ending up with a traceable kind of thing straight back to ontological terms, which can then be automatically added to the Herbenula page, for example, in this case Herbenuli, or whatever page it is that you're annotating. So that's a nice thing, and that's annotating images. And like I say, narrating movies, that's slightly different, but that happens a little bit. We haven't done much of it yet, but it's certainly happening. And I mentioned that we use them in tutorials, and I went through the tutorials earlier, so I'm not going to go over that too much. I'm getting towards the end now. I wanted to show you this, which is a very pretty image, pretty movie, and I will show you this, I think, because it's so pretty. And it's a video which shows really how you can segment things. What I'm going to do is share my screen, and this is the movie, and if we just enter full screen, and I'll just play it. So this just shows you how we have been able to segment things. This is an islet 1 transgenic, and hopefully it doesn't make you all ill when it's spinning around. But you can see we've segmented regions in 3D in a stack, in a double stack. This is from 2 different stacks. This is the trigeminal motor nucleus. I think this is the ocular motor coming out here, and it's really high. This shows you the quality of data we're producing, hopefully, as well, hopefully shows that all very good high resolution as well as in X and Y, and shows you nicely sort of the segmentation. But this is a very slow process. I mean, this took Tom Bracewell, who's one of our workers, a long time to segment, and hopefully we can, if we can sort of somehow automate the segmentation, that would be great. And in fact, oh yeah, that just shows you this, that it truly is segmented. You can actually take out the different bits. I think I'll leave that now and go back. Just go back. I think if I go back here, this is right, isn't it? Yeah, thanks, Anita. I don't know. I think you probably moved me to the right place. I don't quite know how I do that. So we're hoping to adopt this Vibes software, which I think I'm talking about next. Yes. So this is a brand new method which has just come out. We've known about it a little longer than that because we work with this is the Driva lab, and it just came out in Nature Methods, I think, last month. And it's a method to put together kind of models of the where's the pointer gone? There it is. Put together models of the zebrafish brain. So this is a model of the zebrafish brain. This is just diagrammatically represented here, which is created from lots of different recordings of confocal stacks from different angles, etc. And they've probably deliberately been a bit funny with the angles they're using here, just to show that it doesn't matter about the rotation of it. It doesn't matter about whether it's a bit attenuated because it's a bit deeper. And they put it all together, and their software manages to find where every voxel should be, puts together a model of the zebrafish brain using a sort of morphing algorithm, and then will overlay your structure on it. And the kind of anchor it uses is nuclear staining here. So this is the white you see here is nuclear staining, the green is some GFP or something expression. And then you can have an aligned expression pattern. In this case, it's not so much gene expression, but I think transgene expression of the GFP line, but they have done in situ expressions as well, and then you can overlay them if you want as well. And they have a relatively basic atlas, which is particularly basic with regards to the hindbrain, but certainly in the forebrain is reasonably detailed, has a lot of areas which are annotated. And we're hoping to use this system to actually segment our data, and it should be relatively straightforward to use, although it does require taking 4 images, one ventral, one dorsal, from front and back of the zebrafish brain, and even possibly at 2 different sort of illumination levels in order to cope with the attenuation as you go deep into the sample. And that's partly why we're so excited about our new Leica confocal, because it has a resonance scanner and so is much faster. But I recommend anyone who's interested in this to go to this paper. It's really interesting and very beautiful, and has lots of data, supplementary data, which gives you more detail about the sort of what are they called? Mathematics, of course, about how this works, which is definitely beyond me. But fundamentally, it works in a server-based method where you have your recorded data, you convert it to a particular standard format using ImageJ, and you send it to a server, and there's a bit of adjustment that can happen, and then it outputs a kind of standardized brain, morphed brain for you. So we haven't yet had a successful trial of this because it's so new, but we're in the process of doing that this week, actually. So hopefully this is something which is going to really help us annotate our data. And in fact, what we're doing is we're opening up a collaboration with a computer science lab here in UCL to adapt this to be able to work with our already existing large data sets, so that we can use the same kind of the same morphing technology that they're using to do that. Okay, I'm going on a bit here, sorry about that. I don't need to talk too much about this. I think it's kind of covered. This is just to show you really that all this annotation and creation of all of these tutorials can happen online nicely. And tutorials can be edited online, and the idea is that this will be done by curators and editors and people in the community who will be acknowledged over here as an author. Who are these people? Well, at the moment it's us, and we're going to upload all this data once we have the sort of database really complete, and it's getting towards completion now. I reckon in the autumn it will be ready for us to really start uploading things in earnest. And in time we'll kind of involve other people. We have this community page which has details on it. That's from the home page. You can go via here. It also has information about this API which we're developing, which is intended to be able to sort of serve some of this data to other resources, particularly from other species, I imagine, where it might be useful. And then I'm not going to talk about circuits, I don't think. Just to say that we're working on constructs to just delineate individual neuronal morphologies. So that's, I think, pretty much it. I have an acknowledgement slide. We have lots of Cree lines available in Zebrafish coming available. Maybe I'll skip this because I've gone on too long. We're going to integrate vibes. That's one of the main things. I guess that's all you really need to know. And then, yeah, just to acknowledge people. So the Atlas team is Steve, John and Andy in King's College, and Steve, Kate, Tom, Tim, and myself here in UCL, and Stephen, who's kind of he's a sole trader, but he works with us. We had lots of data from Wolfgang and help from Wolfgang, transgenics, and various people. Our funding, most importantly, comes primarily from the BBSRC, which is a UK-based government funding resource. And we have some money from the European community through NeuroExis and ZF Health, and the Wellcome Trust Fund the lab more generally. This is an interesting word cloud that I made from everything that's on there at the moment. You can see quite clearly which areas are well represented, and in et al. It's quite well represented as well. Anyway, just a bit of fun. Any questions?
Okay, well, I think that you've had some applause from the audience, and thank you for doing this. Yeah, my questions, maybe are not pertinent to everyone, but my questions largely involve, the sharing of some of this data, whether or not, how much of this you have thought about. I'm very pleased to see the ontologies basically coming from Zeban being so exquisitely followed, which I think is a really a positive thing, because what you can then do is very easily, dovetail this right into the ZFIN resource in some way that you both decide. But this is a very, very good thing. So and it's, obviously I see a lot of potential here, interactions with NIF in terms of getting link outs from PubMed back into some of these tutorials and whatnot. But I think the real question that I have at this point is, you seem to have at least begun the process of writing what I would refer to as an online textbook, really. And
yeah, right. I mean, you could call it a textbook, call it like a Wikipedia sort of type resource, if you want to put it that way. It's almost like that because we're solicit information, input from other people, but we're just being more heavily curating on it. We don't have the bot and the staff to be able to cope with vandals, so we don't open it up to everyone. But we're effectively it's like a Wikipedia, I suppose, in a way, for the zebrafish brain. But with more data, sort of more anatomical data, certainly. But yeah.
Right. Yeah. So that's perfect. As a teaching resource, what are the licenses? How can I use this data if I am teaching, some students or some such about zebrafish anatomy? What can I do?
In what sense licensing? So, what do you mean by that? So, you can obviously use it as it's a freely available resource. This is government funded. We're not planning on using in making it commercial in any way. So it's just a freely usable resource. Anyone can use it. Is that what you mean? Or?
Okay, but I didn't see, go ahead.
Oh, so this is Marianne. Very nice talk.
Hello.
I know when I'm preparing materials for, say, the medical school, you know, they need, if I incorporate this into a virtual textbook that they're preparing for their students, there has to be certain licenses before you're allowed to do that. Otherwise, you have to get permission. Is that what you're asking, Anita?
Yeah, that's exactly what I'm asking. How can I reuse this resource? What are the licenses that are, you know, that cover this information?
I see. So yeah, it's not something we've necessarily thought a lot about. I suppose we'd be operating on a kind of copyright free kind of arrangement. I don't know exactly how.
You know, the what is it called? I'm always getting it wrong. The Creative Commons Attribution License. So as long as you're attributing.
Right, that kind of arrangement, exactly. We haven't actually considered this. It's a good job you mentioned it, because it's something which we need to consider. Yeah. Yeah, Creative Commons type arrangement, I think, is what we need to get.
So you had. Go ahead, John. Go ahead, John.
If I could just comment in, I mean, I think first of all, the thing that initially drew me to this is just how well designed and how much thought they put into simply just the website alone. I mean, in designing a website that provides so much detail and so many high-resolution images, where you only really get a glimpse of the amount of effort that goes into it by seeing something behind the scenes like this. The website in and of itself is in my opinion, by far the best website to go to if you want to look at zebrafish anatomy, particularly in the 3D sense, and not just standard 2D slices. And I think that in and of itself, from a teaching perspective, is almost invaluable. But I think maybe I could be wrong, but what I'm sort of interpreting the other questions to be is, I don't really think it would be that difficult to turn it into some sort of textbook format on iOS, or even just as an ebook to provide to people as an introductory text to zebrafish anatomy. And while it, you know, of course, there's always more work that could be done and more information that could be added. As it stands now, I think it's invaluable. And you know, I just have to apologize that it took me so long to jump in. I sort of missed it halfway through, but I just wanted to bring everyone together to sort of, you know, help make sure that we promote resources that are putting in the work, and then not just throwing it up on the web haphazardly, but actually thinking about how people navigate and what they can extract and what they can learn from such a source. So maybe if you haven't considered an ebook format or some sort of iOS book format, I think that there's a lot of potential there, given the growing interest in zebrafish research.
That's very kind of you to say those things. Definitely. Feeding back. Definitely. Is your microphone off now? Yeah. Yeah. It's very kind of you to say those things, and I think you might be right. I'm glad to see that someone appreciates what we're doing. Obviously, it's very. When you say it's a great resource, in my eyes, it still doesn't have enough information in there, but that's coming as time goes on. And maybe you're right that developing an ebook type arrangement might be something we could work on for sure. And I'll certainly discuss that with my colleagues for sure. Yeah.
So this is Marianne again. I know we're running out of time and it's late where you are, and we certainly appreciate you interrupting your Olympic
I'm late for my dance lesson I normally go to, but it doesn't matter.
There were some comments that you made in particular about homologies inside the ontologies, and also setting up community platforms or ways of contributing back to these ontologies. And these are two areas that we're particularly interested in. NIF, obviously, I see Jill Boline on here, or Boline on here, and we're working through the International Neuroinformatics Coordinating Facility on establishing what are the best models for dealing with homologies. We sort of balked at a simple declarative of A
yeah,
is
of course you
to
can't
B.
declare that.
Though there is some, yeah, I mean, there's some shortcuts for doing that, and we had some people who advocated it, but I think most of us felt that one ought to be able to do a little bit better than that, and that was really the next stage of where ontology should go. But one of the reasons that we established the NeuroLex was precisely because we felt that the people who are sort of, there needed to be a better interface between the domain scientists who could contribute the knowledge and the sort of structuring that needs to go on to turn them into full ontological terms. And we've now established relationships with like the gene ontology and others to say, you know, we'll review the neuroterms, we'll work on the neuroterms. At some point you will take them into your ontology, but we need a place to sort of work this out in advance. So at some point I'd like to, you know, maybe have a separate conversation to talk to you about that. I know that Jonathan is very interested in seeing how we can better expose the zebrafish terms through the NeuroLex. It is on our list of things to do, just to put the ZFIN anatomy on there so that we have it. But the whole idea is that there are additional relationships and additional terms that need to be added, and not all of those necessarily rise to the rigor that the ontologists want. So that's a slower process, but this is sort of meant to be an intermediary between the two. Also a good way for people to start linking things across domains, because we use the Semantic Media Wiki platform to help put some structure behind the whole thing. So at some point, you know, perhaps we can touch base again on that and how we might help with some of these efforts, and also draw you into some of the international committees, unfortunately, that are trying to figure out.
Yeah, I'd be very keen on being to having those conversations for sure. I think it's I confess to not knowing enough about NeuroLex. I mean, I've looked at the information about it, but I think it would be great to talk about. I'd certainly be keen on participating in the zebrafish kind of certainly brain anatomy sort of parts of it, and yeah, for sure. Yeah,
that would be great. We're starting to get. We've just gotten contributions from Fly. We've gotten contributions from the avian community, the neuroimagers, and what have you. And what we're really working on is this sort of loose community contribution model ahead of turning these things into really formal ontologies. And we believe that there's a place for both of those things that need to go on. So it might be interesting to get used
certainly
as a test case.
And sort of just to take that thought just a little bit further, you know, I approach zebrafish research from a behavioral perspective, particularly from psychopharmacology. And so the way that you've broken it down based on neurotransmitter systems, and eventually hopefully getting into the circuitry of these systems, I think would be extremely valuable, and something that personally I'd like to help contribute to. But I think the overarching goal of what NIF is trying to do is use the semantic web and use the data that's available to start drawing connections between behavior to drugs to brain circuitry to receptors and receptor distribution. And I see this as a way of, at least you guys have the anatomy set very well, and that there's there to push
I
it
agree,
to the next level, and that's what I'm really excited about.
Sorry, I just hear myself suddenly loud. Yeah, it's go from circuits to behavior. I mean, or from molecules to circuits to behavior, and that's what excites us as well. And you know, any contribution we can make to help that happen is really what drives what we do. Yeah. Yeah. Sorry.
Okay. Perfect. Well, I think we've kind of gone past our time, so I I would just like to close by, no, don't worry, everybody does. But I'd like to thank everyone who participated, and I'd like to thank you, Tom. This has been really great, and I think we have definitely some places where we will follow up with you.
Please do.
Okay.
Also, as well, Tom, just so you know, all of these webinars